Okay, lecture 3, we'll talk about nucleic acids. When you talk about nucleic acids, you will probably come up with DNA and RNA, right? So deoxyribonucleic acid and ribonucleic acids are two kinds of nucleic acid we are familiar with. Yes, indeed, we'll talk about both of them. So, what do you know about DNA? So far you've learned a lot about DNA, right? From your previous classes, hopefully. So DNA is what? It's a molecule that, stores your genetic information. You know that. And they are found from where? Inside the nucleus of the eukaryotic cells, right? What does it stand for? Of course, deoxyribonucleic acid is its full name, right? Main functionality, they told you what it is and what it's made out of. So it's made out of it's building blocks. Building blocks are nucleotides. There are 4 different nucleotides, right? G, A, T, C. What does it look like? So DNA is made out of 2 strands. We talked about it in the lecture 1. Watson and Crick strands, and the helical structure, they twist you around. So let's take a look at them. So yes, double-stranded. Okay, 2 strands coming together. So how are they paired up? So bases in them forming these hydrogen bonds between C and G, there are triple hydrogen bonds, and A and T, there are double hydrogen bonds. So if you hold them together, yes, these 2 strands are together. You can see the major group and minor group of the DNA is twisting around. Okay, so it shows that. So wider group and kind of thinner group. So those are major and wider groups. So 3-dimensionally, you see that from DNA. And the backbone here is made out of what? Phosphodiester, the covalent bonds, which is found from phosphate and sugar. Phosphate and sugar, that's the backbone. Okay, and then each strand has the orientation of 5' end and 3' end. So 5' end here, it goes down to 3' end, and the opposite strand should have the opposite orientation, 3' end here paired up with 5' end here, 5' end here, and paired up 3' end here. So that's the structure. Okay, and then the building blocks, I told you, nucleotides. So each nucleotide is made out of 3 important things. Those are phosphate. Okay, so PO minus, and then linked up with the ribose sugar. So it's got 6 carbon, 1, 2, 3, 4. 5 carbons. So 1, 2, 3, 4, 5 carbons there. Okay, 5 carbon sugar. So ribose is deoxyribose. Why did you say deoxy? Because oxygen is removed. Deoxy means oxygen is removed. So only one OH is there. That's the deoxyribose sugar. And then a base is attached to here. So it's one of the 4 bases. A is attached there. So if you see that, that's A nucleotide. So since there's only phosphate, it's AMP, adenosine monophosphate nucleotide. Okay, but there are 3 more kinds: C, T, P. Cytosine, guanidine, G, T, thymine can come here. Okay, so here they are. So adenine, cytosine, guanine, thymine, 4 different bases can come here, right? And then 2 of them are called what? Purine. These are purines with doubling structures, right? And then these cytosine and thymine are pyrimidines with a single link structure. They can come in there. Okay. We learned about that before, so it's a review. And the phosphodiester bond, that's a covalent bond, strong, holding nucleotides together. So phosphate and then sugar, that's a phosphodiester bond linking 2 nucleotides together. Okay, so the oxygen and the carbon, oxygen of the phosphate, and and then so here it's a link. It's a 1 nucleotide here. Phosphate and oxygen. Okay, so there's that linkage. Okay, linkage there. Okay, so that's phosphodiester bond. So phosphate next to the 5 carbon of the ribose in the nucleotide is joined with the 3 carbon OH group of the ribose sugar. Okay, so OH group of the 3 carbon, okay, is linked up with the phosphate attached to 5 carbon of this ribose sugar. Okay, then the orientation of the nucleic acid we said 5' and 3' end, right? Where do they come from, those numbers? So 5' end is right here, which is close to the 5 carbon sugar of this ribose, right? This end. And if you look at this end, so yes, it's 3 carbon of the ribose sugar. 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, right? So that's the 3 carbon close end. That's the 3' end. So that's the orientation of the strand. Okay. Then double strand, like I told you. So one strand here, and then the other strand here on the opposite orientation. Okay. 2 long chains of nucleotides are held together by hydrogen bonds between the bases, and they make complementary base pairing. Complementary meaning what? If you have C there, there's always partnering G, making triple hydrogen bonds. It can go C and G, or it can go G and C. Okay, G and C, right? But if you have A there, you always pair up with T, or T and A. So that's the complementary base pairing. Okay, remember that, right? And then 2 different kinds of bases. Okay, so these are what? Purines. Okay, adenine, guanine, purines. And then pyrimidines are these, cytosine and thymine. These are pyrimidines, single unit structure, double unit structure. Okay. So if you look at RNA instead of thymine, you will have uracil there. But we'll talk about that when we talk about RNA. Base pairing again between purine and pyrimidines. So T and A. So if you have pyrimidine there, it's paired up with purine. So it's always purine and pyrimidine pairing up. Again, purine paired up with C pyrimidine, G purine. Okay. So double versus single, single versus double. That's a base pairing. So because of this purine pyrimidine base pairing, those 2 strands maintain the same spacing between the bases. Okay, because it's single versus double, double versus single. Okay, so distance between 2 strands is constantly maintained. Okay. Tautomeric forms of bases that can induce mutation during DNA replication. What does that mean? It's a kind of unique term, but what it means is, the base structure can be changed a little bit. Okay, once this minor change is done, for example, this cytosine structure is changed from amino form to imino form. It looks like there's a very minor change, right? So hydrogen is moved from here to here, okay, and then so single ring became double ring there. So amino versus imino. It looks very, minor change, minor switch. But from this to this, that change will make big difference when this base is pairing up with another base through hydrogen bonds. Okay, C is normally pairing triple hydrogen bonds, right? But once it's shifted to this, it can only make double hydrogen bonds. So the capacity of hydrogen bonding will be changed, okay, through this tautomeric shift. Because of that, if this minor change is done on your nucleotide, okay, the making of your DNA, so the consequence of this tautomeric change in the next generation is mutation, because normally C pairs up G, but if it's switched up, C will not pair up with G anymore. Instead, it's going to pair up with either T or A. So that's the result. Okay, same goes with G. Normally pairs up with C. Okay, that's the normal form. But if it's switched out to this inner form, okay, so instead of making triple hydrogen bond, you can only make double hydrogen bond from the time on. So that's what we're talking about. So tautomeric shift, it changed the capacity of hydrogen bonding, and that caused a mutation. So understand that. That's what it means. Okay, and base can flip out from the double strand. Normally bases are maintained inside of the 2 strand, right? But they can flip out when methylation happens. So if the bases are methylated and that methylated base cannot form hydrogen bonds anymore, so then instead of staying here, it's going to flip out. Okay, or if there's any damage on the base, and then because of the damage, then repair should be happen. So flip out structure of this DNA is recognized by the enzymes, and then that'll be a cue for the enzymes to do something about that mismatch. Okay, so that's what I mean. So the maintaining the base structure intact is a key to maintain your DNA structure. Okay, that's what it means. And then DNA structure normally exists in this B form. That's what we are familiar with. The minor and major group is helical structure rotating around. It maintains that the same distance, right? But there are some alternative three-dimensional DNA structures. So it's not just a B form. That's what you're saying. Instead, there are alternative A forms or Z form DNA that are possible, and some of these A and Z forms are found identified from chromosomes. Depends on the DNA structure and the modification. Okay, let's take a look at it. So B form, B helix form, is the normal or regular DNA helical structure. So it's rotating around, right? So that's the form we are familiar with. Okay. So this is the B form. So it's the most common DNA form, normally found under neutral pH, pH around 7, or your physiological pH found in the living cell. And under the physiological salt condition, which is about 20 millimolar around. So this is a typical Watson and Crick model, the major and minor group rotating around, right? This is, again, right-handed turn. It's going around. And one turn takes about 10.5 residues, and diameter is about 2 nanometer around the size. 3.4 nanometer is between the base pairs. Okay, a major group is found. So that's a typical DNA structure. However, there are alternative forms which are called A form. So A form, this is really different. You cannot see the major and minor group anymore, right? So this is an alternative form. It's found under the low humidity condition. If water is not there, hydrated, water is all removed, then your DNA will adapt into this form. And this is also found from hybrid between DNA and RNA double strand, or RNA double strand, and then those molecules will adapt A form instead of B form. Again, this is a right-handed turn going around. It's narrower between the bases, and thicker. Of course, this is thicker. Okay, so this is A form. And another form is called Z. Z form is kind of unique form because this is a left-handed turn form. It's formed by alternating purine and pyrimidine bases like GC, GC, GC. It kind of alternates. If you have that unique sequence, yes, your DNA will have this unique form. Kind of strange, right? And the small amount of DNA that exists in this form within the chromosome, and then since this structure is very unique, that unique structure is recognized by other proteins that is functioning in the regulation of the gene expression. So transcription is kind of used as a landmark where transcription factors can recognize this place. It takes longer, 12 residues to make a full turn, and diameter is 1.8. Okay, it's kind of narrower. Okay, so it's a unique form. Z form is an alternative form you can have. So understand three alternative three-dimensional structure of the DNA. Those are B form, A form, and Z form. Okay, so one of the most common form is again the B form. Okay, right-hand turn, as Watson and Crick model, and alternative forms are A and Z forms. Okay, A is closer to B form, similar to right-handed turn, but there's no major minor group typically found from waterless environment or DNA, RNA double-strand formation. But Z form is more unique. It's a very strange form because of that. It's recognized by proteins. It's found only when there's a unique DNA sequence, purine pyrimidine alternating sequence is found, then you will see something like that. Okay, and then